graduate student at the University of Tennessee in Knoxville. He's going to be talking to us about his work in graduate school on mathematical modeling of molecules. So hopefully you're here because you're interested in cells and or mathematics and how things come together. We'll also have some chance questions at the end, hopefully, and a chance to talk to Daniel about his journey into grad school, why he went to grad school and where, and what he's thinking about from there. So welcome, and the floor is yours.
Yeah, I'm Daniel. So let me take a poll here. So who's the biology person? And who's a math person? Okay, we both. Oh, awesome. So the biology is really light. Personally, I don't have a lot of background in biology, so probably the biology teachers in the undergrad probably have taken more biology than I have. So just to give you an idea where I am. So let me lay out the outline here. So first I want to do some basic cell biology just to motivate my project. And then I'm going to talk about special challenges involved with looking at molecules inside a cell, in particular a living cell. And then I want to talk about a particular kind of experiment that's done to measure molecules that are moving inside a living cell. And then I'll address the modeling problem. How do we model this experiment? So let's get into it. So this is a rendering of an animal cell. It's close to what a human cell might look like. Even a plant cell might look something like this. As you can see, there's lots of different structures. You may get familiar with some of them. Of course, the nucleus that contains the DNA, and then there's mitochondria, powerhouse of the cell, the classic name, right? So where's the cell wall? And you may be interested in knowing how do these organelles, the structures inside the cell, communicate with each other? How does the body communicate with the cell? And this is done by molecules passing between the structures, including proteins, amino acids. And so we are interested in these molecules because the molecules moving inside the cell is what determines the cell function. So that includes cell reproduction, cell respiration, and from the point of view of an organism, that's what's going to determine aging, disease, metabolism. So the question of what are these molecules doing is really important to understand what cells are doing and how they're functioning works in the bigger picture. So things we might want to learn would be, what are the molecules? What kind of molecules are they? How fast are the molecules moving in the cell? And that question, how fast are they moving in the cell, is really important to my research because that's important in the pharmaceutical industry to determine drug discovery, how effective drugs are. If you have a drug that's proven to cure cancer, but it moves too slow to be delivered to the cell, it's not going to cure cancer. So really the dynamics, how the molecule moves, how fast does it move, in what way does it move, that's really the questions that I'm interested in. Everybody with me so far? That's basically the biology. Pretty light. So let's talk about how to see these molecules inside of cells. So if you took biology in high school or here, you probably are familiar with this sort of thing. A glass slide, you prepare your glass slide, you put your cell on it, and you might stain it, and then you shine a big bright light underneath it, which works great for certain instances. If you need to see a big area of the cell, and the cell is robust, or the cell is dead already, this works great for certain things. But living cells are kind of sensitive things, and sometimes shining a big bright light on them can kill the cell. Or maybe it's not that drastic. Sometimes that big bright light under the glass slide, it'll just change the cell's function, and you're not going to be able to answer the questions maybe you're specifically interested in. And then even on a chemical level, shining that big bright light or whatever you have to do to prepare this glass slide can change the chemistry of the molecule that you're interested in. And so the idea of how do we measure the molecule inside the cell with minimal disturbance to the cell function and to the chemical properties of the molecule? That gave rise to a different kind of microscopy than this glass slide wide field microscopy. And it's called confocal microscopy. And the whole idea is to just send as little bit of light as you can. Basically, just light at one little point inside the cell and see what happens at that. So this is the idea. You have inside the cell a molecule floating around, and you just have one little point of light. So your molecule's floating around, and as it gets closer to this point of light, it's going to be emitting more light. And when it's farther away, it's going to be emitting less light. And the way that this works in practice is you have to prepare these molecules to fluoresce. And so you bombard them with a certain wavelength of light, and then they emit a different wavelength of light. And so you're just trying to pick up the light that the molecule itself is emitting. So this is the type of microscope setup that I'm interested in. This confocal microscope setup, where I'm just lighting up a little point of light inside the cell. And again, the whole idea of this is that it prevents a phenomenon called bleaching, which comes from shining a bunch of light on your cell. The bleaching can damage the cell, change the cell function, change the molecular properties, and the behavior of the molecule may not be able to be captured. So this is how you try to measure molecules in a living cell without changing their behavior as much as possible. It's shining this little bit. There's lots of technical details about how they do this, and you can incorporate that into a model. And like a lot of modeling, you get to a point of diminishing returns. When you put more and more detail, closer and closer to reality, your model may be more and more accurate, but analyzing that model may become intractable. So this is kind of a basic trade-off with your model. You want to have a parsimonious model where it's close enough to reality, where you can learn something about reality, but not so close to reality that it's as hard to learn about how reality is. So you want to simplify, but not too much. Okay. So the next thing would be, okay, so we've got a molecule floating around, it's getting close to the source of light. So what kind of measurements does that produce? So in reality, you have biologists in the lab, they've got this confocal microscope, and they're trying to learn something about these molecules, and they're going to get some kind of data. And what kind of data that is determined by the technical setup of the microscope. And so the idea is you can think of, so I want you to think of the molecule moving around, and as it gets closer to the center of brightness, it's going to be emitting more photons, and that's what we're picking up here. So what this graph down here represents is how many photons that the molecule is emitting. So I call it an average photon count, and that's for technical reasons that I'm going to sweep under the rug. If the biologists are interested in that, I can talk about it at the end. So Watch the motion of the molecule and see if you can relate it to this graph output. So as time goes from left to right, this is going to try to capture how many photons are being emitted from this molecule. So it gets close, and we have this first peak, and it goes away, it sinks down to the valley, gets close again, but not as close to the center. And that's the idea. We want some kind of function of how close to the center the molecule is. So this sort of represents the photons being emitted from the molecule, which is not what you can measure. So let me run this back one more time, and I'm going to ask you to do something with it. So I'm going to ask you to close your eyes and open your eyes like a camera shutter, and think about what that means for measuring where this molecule is in the cell in relation to the cellular brightness. Okay, so start with your eyes closed and play. Okay, open your eyes, close your eyes, open your eyes, close your eyes, open your eyes, close your eyes, open your eyes. So this is really how this machine works. It's kind of working like a camera that's opening, collecting the photons, and then it closes its eyes and spits out a measurement. So if you think of it like this, as time goes on, the camera opens its shutter, collects the photons, and then shuts its shutter. It opens the shutter, and then it shuts. It opens the shutter, and then it shuts. So this is how the measurements are collected in the focal microscope setup. And this particular type of measurement is called spectroscopy. There's other ways to use a confocal microscope, but this is the particular type of experiment that I'm interested in. So the idea is you collect for some time period, and you add up all the photons that you counted, and that gives you the first measurement. Collect for another time period, that gives you another measurement. And you collect for another time period, that gives you another measurement. And so really what the output of these confocal microscope spectroscopy experiments are is data points like this that give you a photon count, which represents the accumulated photons during one time of exposure for the shutter measurement. So you can see we're starting to get a pretty coarse idea of what's actually happening with this molecule, right? It's kind of a pretty distorted information of what's actually happening with this microscope. It's moving through the cell. This just tells us how close to the center of brightness it is. And then we have this extra layer of distortion from the camera opening and shutting. And then within a single exposure time, that whole thing is just collapsed to a single point. It just adds up all the points. And so this is the type of data you get from these experiments. And what we would like to model is this data. How do we simulate this sort of data? So that's the experiment. Feel free to interrupt me at any time as well. That's fine too. If you need me to repeat anything or go back, I'm happy to do it. Or explain something more. So that's the experiment. Now we want to model this experiment. So we talked about a few things that we need to address in the model. Number one, the motion of the molecule in the cell. And the way that we model the motion is with a random walk. And I'll explain more how that works. And then we have this brightness function. How do we model an area, a point of light in the middle of the cell? We use a particular kind of function called a Gaussian, and I'll explain more about that. And then finally, we have this photon accumulation step, and we need to figure out how to magnetize that. So really, there's things happening in the molecule, in the model, things happening in the microscope within the model, and then the measurements that we do. So let's talk about the molecular motion. So we need to give a name to the initial position of our molecule. We'll call it X naught. And our molecule is going to move randomly to a new position, X1. It's going to move randomly to another position, X2. And then randomly, and so on and so forth. So it's kind of bouncing around. And in fact, the random walk is already an approximation to how the molecule is moving, because the molecule moves more closely to something called Brownian motion. And this is some math that was developed by Einstein by studying how pollen moves in a suspended liquid. So it's a random motion, but it's a continuous motion, right? So as the molecule is moving from the cell, it's not jumping from one point to the other, it's continuous. So we approximate that with this random walk, which is a discrete process. We are only going to know the position at X0, X1, X2, and so on. So let's break this down a little more. So we have our initial position, X0. And if we know our initial position, how do we get our next position? Well, we take our initial position and then add a random step, and that gives us our next position. Okay, and we just proceed like that. To get our X2, we just take our X1 position, add a random step, and that gives us our next position. So this random step is doing a lot of work. This really contains all the information. So let's dig into that a little more. So let's call R this random step, and we got to answer a question. What kind of object is R? So we model R by a normal random variable. So what this means is that it's really unlikely that from one step to the next, that the molecule is going to make a large jump in either direction. It's more likely that the molecule is going to take a small jump. But we want to allow for the molecule to stay put. We'll just move a little bit, we'll move a little more, and somehow encode that it's really unlikely that the molecule is going to dart from one end of the cell to the other. And we do that by this type of probability distribution. So we just take a sample out of this probability distribution, and we add that step by step. We have our initial position, we take a sample, which gives us our random step. Maybe it moves a little to the left. And now we've got a new position. We take another random sample, maybe it moves a little more to the right. So by iterating this random walking, that's how we model the molecular motion. And we can make this more and more accurate by taking the steps smaller and smaller. We can make it close to Brownian motion in a sense by taking these random steps smaller and smaller. So we have some freedom here. So I said it's unlikely that the molecule is going to take a large step. But of course, molecules behave differently depending on the chemical properties of their function in the cell is. Some molecules move faster than others. And we can actually use this random variable to model that. So this blue curve and this orange curve are both normal curves. They're valid distributions for our random variable. And we might pick one over the other depending on the properties of our molecule. So if our distribution is peak very high, that means most of the probability is close to 0, which means most of our random steps are going to be small. So we call the amount of being peaked the variance. If it's very peaked, it's low variance. So if you're concentrated close to 0 with low variance, that's how you model slow. That means your steps are most likely to be small. If you want to allow for bigger steps, then you need more area farther out in the tails. And that relates to a high variance normal distribution. And that's what we use for fast. So in practice, this is actually the thing you want to learn. How fast and how slow is the molecule moving? Because that can tell you what kind of molecule it is. And so I can talk more about that. That's beyond the modeling step to an inference problem, but that's a big part of what I work on too. So let's talk about the confocal microscope. How do we model this brightness spot? So let me just give some names to things. So we'll call X the position of our molecule, and C is going to be the center of our brightness. So we've got our space that's inside the cell. We're sending a point of light into this cell. Again, we do that so we don't damage the cell or change the chemistry of the molecule. And we'll call the middle of that bright point C. Okay. So we want some kind of function to model this brightness. So I'll call this function b of x, and we want it to have certain properties. We want it to have a high value if x is close to the center of brightness C, and a low value if x is far from the center of brightness. All right? And it may shock and amaze you, but this is actually going to work very well if we use another function shaped like this, right? So if x is close to C, this will be a high value. If x is far away from C, it's going to be a low value. And brightness, we would normally model it as a positive value. And so we wouldn't want this thing to pass below the x-axis. Now, there's something about the symmetry here as well. So we want, if you're the same distance on the right or the left, you should be the same brightness. And so we want this function to be symmetric as well. And there's a reason why the same function describes this and the random step in the random walk. And it has to do with how these types of distributions are related to circles. So let me kick it up a little bit, because obviously x is one-dimensional here, which means our graph is two-dimensional, but our molecule is living in three-dimensional, right? So first, let me give you the functional form. Oh yeah, let's make a simplifying assumption. We can choose our center of brightness to be 0. As modelers, that's okay to do. We can just change our frame of reference and give them names to things. As long as it's truly just renaming things, that's always going to be valid as model. As long as you can translate back to the original names, that's safe to do. So we'll assume that our center of brightness is 0, and that our brightness is high if x is close to 0, and low if x is far away. So in that case, we can take a functional form for b to be a 1D Gaussian. So that has this form. We take b of x equal to e to the x squared over a. And this a is a parameter that we can play with. The a is what controls the variance, or how sharp this peak is. In other words, are we very bright at a small point and then fall off very quickly in the brightness? Or does the light diffuse more slowly? So we can use A to control that. So like I mentioned, in this diagram, X is one-dimensional, so the graph is two-dimensional. So there's a higher dimensional analog to the Gaussian. So we'll just build up step by step. So if your molecule position is now X, Y in 2 dimensions, then we'll need a 2D Gaussian function for our brightness function. So in this case, we want when X and Y is close to the 0, we want a lot of brightness. And when X and Y is far from 0, we want the brightness to fall off. And as you can see, when our input is 2-dimensional, our graph is 3-dimensional, actually. So the function, the Gaussian in 2D looks like this. So it's similar, but now your exponent has an x squared over a plus a y squared over b. So in 1-dimensional, you just have this, but in 2-dimensional, you have this. And now we have more parameters to play with. The a allows us to squeeze and stretch in the x direction, and the b allows us to squeeze and stretch in the y direction. So I mentioned that these Gaussian functions, these normal probability distributions, come up because of something about circles. And the idea, you can see it now that there's more dimensions. If you travel, say, along the same height on this graph, you're going to trace a circle. And that's a property that we want. We want objects that are the same diameter away from the center of brightness to give us the same brightness value. And so that's why this is an appropriate function. And the same goes for the random walk. We want the same likelihood as to go in any direction. And so that's why we have the same function. Okay, so let's get real now. So the molecule is actually living in 3 dimensions. The molecule's position is described by an x coordinate, a y coordinate, and a z coordinate, right? So that's really what's happening. So we really need a 3D Gaussian function. 1D Gaussian function gave us a 2D graph. 2D Gaussian function gave us a 3D graph. So a 3D Gaussian function gives us a 4D graph. So I can't draw that. I couldn't find an image of it. But here's the functional form. Now we have 3 inputs, each of our coordinates. And it's going to look just the natural extension. You just add one more exponent for your z squared. And now you're able to squeeze and stretch in the z direction as well. So as far as visualizing this, all is not lost. Because sometimes it's easy to get lost in the details when you're doing math. What are we doing here? This is a brightness function. Right? So really the way to think about this is like light bulb. And it's bright near the middle, and then it gets dark as it goes away. And so you can kind of think of it in that sense. So that's what this brightness function is about. And this is, in fact, what we're using in our model for the confocal microscope. This is how we model that brightness in our confocal microscope. Yeah.
Wondering why do A, B, and C and not just the same parameter and the denominator there?
That's a great question. So the question is, why do we need A, B, and C and not just have the same stretching or scaling in each coordinate? So if we had A equals B equals C, then our brightness function would kind of be spherical. As you moved away from your center of brightness, if you were on a spherical shell, it would be the same amount of brightness. So the confocal microscope is actually set up to where A equals B and C is different. So actually the shape of the bright spot in a confocal microscope is kind of egg-shaped. So it has like cylindrical symmetry, where you have a circle that gives you the same brightness, but it's more stretched out along the z-axis. Similar question. And that has to do with technical details about how this microscope is built and also about the physics of light at such a small scale. So these are some details that I swept through a little bit. Okay. So let me just remind you of this. Now we have some math to attach to it. So remember, this is just describing our brightness function, right? And so this is supposed to tell us the photon emission of our molecule as it moves around the cell and gets closer and farther from our bright spot. So the question is, how do we get something like this mathematically? We want something like this, and so we have to model it. Now we have a piece, we have our brightness function, and we have our molecular dynamics, right? So let's talk about the measurement step. How do we accumulate the photons in our photon detector? How do we model that? So our molecule is moving to the cell, closer and farther from the center of brightness. It's described by x, y, and z coordinates. Those x, y, and z coordinates are changing over time according to a random walk. So x, y, and z are actually functions of time, right? And our brightness is a function of x, y, and z, right? So if we want to, basically, we want to be able to plug in our molecule into the brightness function and say, how bright is my molecule? That'll tell us how close to the center of brightness it is. And so since x, y, and z are changing over time, when we plug in the molecule, we're going to get a brightness function that's changing over time as well. So really what we want to do is define a new function that keeps all this information in one place. And that's what our average photon count function is. We just call it U. It's a function of time. And really it's just renaming our brightness function, but now we're feeding in a molecule that's moving through the cell. And so we plug in a certain time and it spits us out how bright is our molecule. How close to the center of brightness is our molecule. Everybody good on that? And so really this is what the graph is describing here. It's telling us how bright a molecule is at a certain time. How close to the center of brightness it is. All right. So let me bring back this diagram. So we've got all the way here, right? So we've modeled all the way up to this point. We've got a way to find average photon count with our function U, which is like plugging a molecule's position into a brightness function. So what's left to finish our model, before we model this experiment, we have to model this step where the aperture of our photon detector is opening and closing. And then we have to model the step where it's adding up all the photon counts in these measurements. Okay. So we need to specify a couple more things here. So our photon detector is opening its eye and closing its eye, opening its eye, closing its eye. And when the eye is open, it's accumulating photons. And then it adds up all those photons and gives us our nth measurement, Wn. The Wn is the data that we actually get out of this experiment. So one thing we're going to need is some name for how long the apertures are. So let's call that S. This is going to be the length of time, and it just tells us how long the apertures are. So that's going to be important when, to model what Wn actually is. So, and again, just a reminder, this graph, that's our average photon. That's telling us how bright the molecule is. It's approximately how close to the similar brightness this molecule is, which tells us something about the brightness of this molecule for the cell. All right, we're almost there. Once we have an expression for our measurements, we reached our goal. We wanted to model this experiment. If you model the measurements, you model the experiment. All right, so S is how long that aperture is open. So the aperture opens at some time Tn, and then it stays open to time Tn plus S, right? And so this interval Tn to Tn plus S, that is the interval on which we're collecting photons for our information. Okay? So if we want to collect, let's look at our last measurement here. We want to collect photons according to this brightness function, add them all up, and spit out a measurement. So can anybody guess the last step here? What do we do if we want to add up all the values in the function? Integrate. Exactly. So we have our function u. We know we want to add up all the values during the time that our aperture is open. So we model that with an integration. So we model our measurement, Wn, by integrating over this time interval, which represents when the aperture is open, and we integrate our brightness function. So at each time, it's telling us how bright is a molecule. And so we accumulate all during this time the photons bouncing off our molecule. And that's how we get our last measurement. And so we model the experiment. So I swept some stuff under the rug. If there's any details that you want, then feel free to ask. So thank you for listening. Let me do a quick summary. I said cell biology is important because that determines cell function. That's important with respect to disease or immunity. We wanted to limit bleaching, so we couldn't use our standard microscope setup. So we had to come up with this way of just shining a little bit of light in the ceiling. So that was our confocal microscope. And then we had this idea of the camera opening and shutting and accumulating the photons to measure the motion. And that was our spectroscopy setup. And then we just modeled each of these ideas. So we did the molecular motion with a random walk. We did our brightness function with a Gaussian. And then we used integration to model our photon. Okay, thank you so much.
We're tracking this research and what you think the most important implications of this research will be.
What was the first part?
What drives me to do this research?
So, as often happens in grad school, you don't get to pick exactly the project that you're working on. So, my advisor is Dr. Johannes Stralis in the UT Math department. And this is related to some research that he's been doing over the past 7 years. And, you know, I could have refused, but I found it interesting. So he said, "Are you interested in this?" And I said, "Yeah, that seems cool." And you said, "Why do I think it's important?"
What are the most important future implications?
Okay, so I think the main thing is, well, when you model stuff, that's the first step. You know, some people focus on that, but in my project, that's just my first step. And the reason that I want to model it is because I want to use it to learn something about the the real life experience. And so I have to use statistics to go back to the real life problem. So this is what they call the forward problem, the modeling. And then the inverse problem is to say, okay, given some measurements, what can I say about how fast the molecule is moving? Or what can I say about what types of molecules will generate such data? You know, what number of molecules or proportions of molecules will generate such data? So those questions are important because if you can learn about the molecules that generate a certain kind of data, then that can tell you why a cell is functioning a certain way in relation to the molecules inside the cell, the dynamics of those molecules, concentrations of those molecules, and that you can learn about disease and aging, you can learn about cell function, metabolism, cell reproduction. You can learn lots of different real-world applications. You think about it in the reverse direction you do every day, where you start with the measurements, then you get back into it. But what does that say about the cell?
How long is the aperture of the photon?
It's on the order of a second.
Oh,
Yeah. So, and that's because the light is very dim. So you're actually not collecting very many photons. A single molecule, most of the time, you would not collect any photons. It would be too far away. And it's moving randomly, so you'd be lucky to learn something if you're getting 4, 6 on the order of 5 to 10 photons per second aperture opening.
so you keep track of like one molecule at a time?
So yeah, that's what this research was originally designed for. Was to study single molecule dynamics. Yeah, and which is reasonable in a cell. The size of the cell compared to the molecule is huge. So the molecules, even though there's a lot of them inside the cell, they kind of live solitary lives. So single molecule dynamics can be important. So,
Are there differences in as far as like recording light from molecules with different membrane composition? Like, is that a factor that has to be considered? If like lipid composition was like 70% versus 50 or something like that?
that's an interesting question. So, I think it's generally, so there's a way to prepare the molecules such that they fluoresce. And there's only a few ways to do that. And so typically you're shining blue light on the cell and you're recording green light. So I would guess that since the kind of fluorescence technology is kind of limited, there's only a few ways to do it. Yeah, there's probably things you just can't measure depending on the certain structure. And actually the most recent Nobel Prize that just came out yesterday was about this stuff called click chemistry, which is about a new way of fluorescing molecules. So this is still a really active area of research.
Thank you.
Are you learning anything about how molecules move differently in different environments, like inside the cell versus outside the cell, or will you consider that as a research question?
So this confocal microscope setup is specific to measuring inside the cell, what they call in vivo, in living cells, and it's because it's not shining much light on the cell. So the whole idea is that we want some kind of microscope that won't disturb the cell very much. So if you're outside the cell, you can be a little more aggressive. You don't have to worry about killing the cell or changing the chemistry of the cell.
Do you think your research could maybe contribute to like public health and cancer research?
I hope so. That would be great. Yeah. Yeah, I mean, theoretically, anytime you learn more about molecules in living cells, that could lead to applications in drug discovery, could lead to applications in disease management. That would be great. Make me feel good.
Give maybe just a few examples of molecules you've used in the research.
So proteins mainly, and actually I've not got any data from a lab yet. So I'm still in the kind of simulation phase of things. And I'm trained more in pure mathematics, really. So the application side of things is still new to me. So really, I'm always going down rabbit holes, mathematical rabbit holes, and I think that eventually we do have a plan to get to some wet labs at UT and get some data. But there's no real world here yet. It's just a real issue. Yeah. Thank you. Mm-hmm.
So most of what goes inside the cell is really organized and it's not based on gravity. So how would your modeling change to add that component, which is more realistic to what happens in the cell?
Yeah, so like the pathways that the molecules might take. So we had to change the function of the random walk. There's some randomness, right? But then a Gaussian wouldn't be appropriate necessarily. We want some kind of function that incorporated this directed movement, but still allowed for some kind of wiggling. And so that would be in the modeling step. You'd have to change your random walk function to have there's ways to constrain them to certain pathways. And so as far as the reverse problem, it might be interesting to say what types of pathways might give rise to these measurements. And that's a question that you could set up as well.
And could you take a few minutes just to tell us about your path from undergraduate to graduate school, where you go to undergraduate, how you decide to go to grad school and decide on?
Yeah. So I'm in my third year in the PhD program. I did my undergraduate at UT as well. I'm from Knoxville, born and raised. And I actually never took a derivative before I was 30, and just got into, it was really YouTube that got me interested in science and mathematics. I'd come home from work and watch Khan Academy videos and read them around and stuff, and decided I should just go back to school, and I did.
Would you mind saying what kind of work you got into, or what you did as an undergrad?
Yeah. So when I did a rushed curriculum at Pellissippi State through all the like first and second year math stuff. And when I went to UT, my advisor was one of the senior algebra faculty, and I said, "Well, this is great. I want to do algebra." And he said, "Okay, what do you want to do afterwards?" And I said, "It'd be great to do a PhD and work in a lab at ORL or something. I'd love to stay in the region." He said, "Well, you can't do algebra because there's not jobs in DC or be an academic, and in that case, you can get picked where you want that." He said you should study numerical analysis and applications. You will get a job at ORL. And I was game to do that. I didn't know what that meant, but I grew to love it. It's really interesting stuff. And so that's how I got into the applied field. Really a pragmatic concern of, I want to stay in the region. And I did find stuff that I liked.
So I hopes one day to be at a place like L'Oreal or. Fine.
Yeah.
Any other questions for? Thank you very much again. Thank you, everybody, for coming. Have a great day.